All right, here we go. This is like your lesson for today. And I'm sorry, I had some technical issues there in third period. You guys should have really told me if you were in third period on the Teams meeting that you didn't see the actual slides changing. Might want to let me know on that next time. I guess I clicked share on the wrong one. I apologize for that. So sorry, but let's talk about section two. And in section two here, we're going to talk about photosynthesis. So we already started talking about photosynthesis a little bit. I talked about how these are relating together and how they're so important for us. So, like for example, on this overview of photosynthesis, we talked about how autotrophs, what they'll do is they will make organic compounds using photosynthesis. So they can actually make and store up energy molecules, and they'll do this in a process of photosynthesis, which you've heard of before, which is just converting light energy into chemical energy. And this right here is the overview of. This is what you see. So they'll take carbon dioxide, which we exhale out, combine it with some water molecules and light coming from the sun. They will take that to make sugar molecules that they are storing energy within, and oxygen, which is good for us because we breathe the oxygen in. And this is like the good trade off for us. Okay, they give us what we need to breathe, and we give them what they need for them to be able to produce their energy molecules. So within this, it happens within two phases. The first one of this is the light dependent phase. This is where light energy is converted into chemical energy. And the second one is the light independent phase. We're going to talk about both of these. And this is where chemical energy is used to synthesize our sugar molecules, and glucose is an example of that. So within this, we have phase one, which are our light reactions, and that is where the absorption of light is the first step in photosynthesis. And what happens is once light energy is captured, it can be stored as ATP or NADPH. So within this, the organelle, which you guys should know at this point, that is allowing this to take place is the chloroplast. And this is where the light capturing pigments are held. This is where the light is captured and the process of photosynthesis will take place. And the two main compartments and components of this is the thylakoids, which are flattened sac-like membranes, and they are stacked, and they call those granas. And the fluid filled space outside the grana are called the stroma. Now, in this, different pigments mean different things for us. Okay, the pigments determine which frequency and wavelength of light the plant will absorb. So different pigments absorb specific wavelengths of light. So as you can see here, you got chlorophyll A, chlorophyll B, and carotenoids. So in this, chlorophyll A will absorb blue light and it will absorb orange light, but that's it. Chlorophyll B will absorb blue light, and that's it. This is where, like grass, is green. And it makes sense because the colors that we see is based off of colors that are absorbed and refracted off, reflecting off. So let's say you have a blue shirt on. The reason your shirt is blue is because the shirt is absorbing all other wavelengths of light except for blue. It will not absorb that wavelength of light, so it reflects it off, and that is what we see. We get to see the blue because it's reflected off and not absorbed. So whatever pigment you see, that means it's not going to absorb that color of light because that is the color it's reflecting off. Within this, you have the phase one, which is light dependent, the light reactions. So this happens in a process of electron transport. So the first thing that happens is the thylakoid membrane has a super huge large surface area, providing space for a large number of electron transporting molecules. It's hard to roll off the tongue today. And two types of protein complexes called photosystems. So you have photosystem 2 and photosystem 1. So in this is where they house the light capturing pigments for this process. So within this, the light energy excites electrons within photosystem 2. So right now we're focusing in right here. And what will happen is the light energy causes a water molecule to split. So you can see it here. We got a water molecule, and it splits. And it will split, releasing an electron into the transport system. So here's our electron. It releases out a hydrogen ion or a proton because it doesn't have the electron now. The electron's loose. Here's the hydrogen ion or the proton, however you want to call it. And it also gives oxygen as waste, and they just kick it out because they have no use for it, which is good for us because we have great use for it. This is the first step. Now, in this, what happens is the activated electrons will move from photosystem 2 to acceptor molecules in that thyroid membrane. So here's the electron. It comes in here, and it'll move through here in these electron carriers. The electron acceptor molecules transfer the electrons through along the series of electron carriers to photosystem 1, which we can see right here. Now, the important thing of it is we have these hydrogen ions building up on the inside here, which is important and is going to come into play later on. Remember about these hydrogen ions building up, concentration gradient on the inside of that molecule, on the inside, not molecule, the inside of that organelle. So in the presence of light, we're still in phase 1. Photosystem 1 will transfer the electrons to a protein that we call ferredoxin. So here's ferredoxin, the final electron acceptor. And the ferredoxin transfers the electron to the carrier molecule NADP+, forming the energy molecule NADPH. So NADP plus hydrogen ion come together with electron to form NADPH, which stores energy. And this is what I was talking about, that hydrogen ion concentration gradient. Remember when we talked about ATPase pump? When we talked about them, it was all about building this huge concentration gradient so that it would naturally flow and things could flow with it. Because we built up that hydrogen ion concentration gradient on the inside here, now what is happening is we can have another electron transport taking place here in conjunction with it through chemiosis. Through this process of chemiosis, ATP is produced through the flow of electrons down a concentration gradient. The breakdown of water molecules provides the necessary protons for the ATP synthesis system to take place and to happen. That's what I was talking about with these hydrogen ions getting built up and that concentration gradient building up in there. So in here we have the second phase, phase 2. Now this is light independent. So light doesn't matter at this point. This, excuse me, this is the Calvin cycle, okay? In the Calvin cycle, there's this big huge thing. I'm just going to give you a general overview of this thing. I'm not going to require you to memorize everything, every step. I will openly tell you if you're going to become a doctor or a pharmacist or some scientist, you're looking at taking Biochem 2, at which point they're going to make you memorize the Calvin cycle, every part of it, inside and out, and what you get out of every step and every phase. So in this, sorry. The first step of the Calvin cycle is called carbon fixation. So what will happen here is we'll take CO2 molecules and they'll combine with 5 carbon molecules to form 3-PGA, phosphoglycerate. So this is the first step of what will happen. So we're starting with some carbon dioxide that will combine together and we'll end up with 12 3-PGA. In the second step, chemical energy is stored in ATP and NADPH is transferred to the 3-PGA to form glyceraldehyde 3-phosphate, or as we like to abbreviate it, G3P. So now we have 12 G3P setting here. In the third step, some G3P molecules leave that cycle to be used for the production of glucose, and other organic compounds. Some of these, for example, is fructose and starch, and there can be others. From there, the fourth and final step, an enzyme called rubigase, converts the remaining G3P molecules into 5 carbon molecules called ribulose 1,5 biphosphates, or as we abbreviate it, RUBP. These molecules combine with new CO2 molecules and continue the cycle. So literally, we're feeding the cycle here. This feeds the cycle. Now, within this, there are some alternative pathways. Like, for example, there's such thing as C4 plants, and the C4 pathway allow plants to maintain photosynthesis while reducing water loss. This is very special for certain plants if they don't want to lose a lot of water. Significant structural modification in the arrangement of the cells within the leaves separate the CO2 uptake from location of the Calvin cycle. Cam plants, for example, are found within desert plants. They collect carbon dioxide at night and store it into organic compounds, and during the day release the carbon dioxide from the organic compounds for the light-dependent cycle of photosynthesis. And that is the end of section 2. Make sure you get your notes. Make sure you got these down. I'm sent the PowerPoint along with this video. You should have both of them. And besides that, I will see you guys as soon as we get back to school. Peace.